So I'll try my best to sort of convey, I have two aims today. One is that to convey what is exciting, right, about bacteria. This will be a little bit of a biased viewpoint, of course, because it'll be my viewpoint, right? And it'll be the viewpoint of people who've worked with me, my team members, and so on. But I think it's also really incredible to know where this field is going. So I hope that in the end we can also discuss a little bit more what it is that is important about this work, that in this field, and what more can we learn in the coming years, because this field has just started exploding. So this picture here is a phylogeny. It's a tree of life. So a tree of life shows all the creatures of the world, right? And what we're trying to do here, though, is in this tree, we're trying to understand something about their relationships to each other, right? So maybe some of you have heard about this, where these fingers here in this part of the tree of life each finger represents one species, right? So one type of organism. And this particular bit of the tree of life, this is just one large branch, belongs to the bacterial clade. And this, of course, is not really showing all of the species that exist, right? This is just a few, like a very small fraction of the types of bacteria that we actually know exist in the world that we have found. We as in, I'm talking about the larger group of scientists who studies these, right? So, so each of these little fingers at the end is one type. And then all of this big cluster of fingers here represents all different types that are very closely related to each other, right? So they shared a common ancestor in the recent past. Similarly, this group goes back and joins up with this cluster here. And so here is where all of these types shared a common ancestor, right? And why is this important? Because it tells us where things came from, right? How things have evolved. And therefore, what traits might they share, what they may not share, and so on and so forth. So this is a very, very fundamental framework in which I think all of biological knowledge can be organized very beautifully. And it really is important to give us a lot of insights. So I won't talk about trees too much, but I want to talk about this a little bit more because I want to orient you to what I'm speaking about. So as I said, this is the bacterial part of this tree. And this, by the way, is are pictures that I'm going to show you just now are from the Microbia Museum in Amsterdam. Okay, so here are the bacteria. And then now you can branch out from this big bacterial branch into another set of organisms which are also single-celled and tiny, like bacteria are. And these are the archaea, okay? And then yet another big branch is Eukarya. And Eukarya includes all of us in this room, all the plants, all the animals, right? These are all what are called eukaryotes. And what is the distinguishing feature of eukaryotes? We have sort of larger cells typically. We typically, although not always, are multicellular, which means we have many, many, many cells in our bodies that work together, right? And these cells themselves are much more complicated, right? They have many different pieces inside them, and all of those little pieces are compartments that work together. So think of, for example, something like a city, right? So we need, you know, the water department, we need the electrical department. All of them are units that work together to make the whole lot function. And that's kind of what's happening inside these complex eukaryotic cells. And then they come together to form a big organism like all of us, right? So eukaryotes are sort of larger, bigger. Bacteria and archaea are smaller, single cells. We usually can't see them with our naked eye. But what's really astonishing is these different major branches of life have come together and have interacted millions of times, right? And continue to do so. And that's what the goal of my talk today is going to be. And by the way, just for, this is where humans are, at the very corner here, right? And this is not drawn to scale. So there are many, many, many more bacteria and types of bacteria than there are eukaryotes, right? But just because we have a very eukaryote-centric point of view, we are more interested in humans and animals and plants that we observe and see easily, interact with every day. We don't realize how much we're actually interacting with all of these little guys here. Yeah. Anybody have any questions so far or a comment? No? Okay, all right, yeah, question.
Bacteria and archaea cells are less complex because they're not eukaryotes, they're prokaryotes.
Talk away from me. Yes, Absolutely, because they're called prokaryotes, right? But that's why we call them prokaryotes and eukaryotes. It's not that they are and therefore, but we call them there. You're right, yes, they're prokaryotes, right? So they're basically primitive in some sense. Okay, all right, so let's move on. I want to show this as a start because I really like the view that this gives, that this is a galaxy unto itself, right? What are all of these points here? Each little dot represents a sample which somebody has gone out and collected. Maybe it's a soil sample, a sample of seawater, or guts of vertebrates. Vertebrates are things with backbones, right, including us. So each dot here is representing a clustering of many different microbes, right, bacteria typically, that were found in each of these samples. And what they've done then is simply represented these clusters, again, in a fashion somewhat similar to the tree that I showed you earlier, where related dots, which are most similar in terms of their composition, are close together, right? And so there are little lines joining all of these which you can't see, except in this part, which is all red. So these sets of samples are very, very similar to each other, okay, in the kinds of bacteria that they have. And that's all this plot is showing. But what I love about this is it's beautiful to see how nicely the vertebrate gut samples separate out. The seawater is quite distinct, soil is quite distinct, and there are a few other kinds of samples here that I'm not going to bother with, right? But what this tells us is, broadly speaking, the kinds of bacteria that live in the soil versus seawater versus our guts are quite different from each other, right? And that alone is interesting. Why is it interesting? Because it suggests that there's something more going on than just, us passively getting these bacteria. So for example, I think all of you have seen little kids playing in the soil and the sand, and sometimes putting stuff in their mouth, right? So if that was how we acquired all our bacteria, then as a child, our gut bacteria should look very similar to soil bacteria, right? But they don't. And what that means is our guts are very good at also filtering out a lot of bacteria, not letting them survive, not letting them live. And that's what happens inside our guts as well. So I'll talk a little bit about that too. So in general then, this is what this world, this explosion that I'm talking about is leading to. It's an enormous amount of data, because each dot here, remember, represents hundreds of types of bacteria. And we still haven't figured out what all of these mean, right? What do all of them do? What's also needed as a result of this explosion, by the way, this has come about because of new methods of sequencing, of DNA sequencing. Who's heard about DNA sequencing? Oh, good, fair number, but some have not. So I'll quickly explain what that means. You all know that our genetic material is DNA, right? And what that DNA is essentially a bunch of 4 letters that have come together, right, in a string. And one wants to understand what letters are there, in what order, how many of them, and so on, right? So it's a bit like reading a book where you have letters, but you have words, you have sentences, and that's where the meaning comes from, right? So without that, you would just have a gibberish, a string of letters. It wouldn't make sense. So if you want to understand sort of fundamentally what makes us what we are, it's the DNA sequence, right? Of course, on top of that, it depends what you eat, it depends on what you learn, a lot of things happen. But fundamentally, our DNA is important. And so we've been trying then, as scientists, to sequence the DNA of everything we can possibly sequence, right? And what that involves is essentially reading these letters, right, and pulling them together. Now, the problem is that we have very little DNA to work with very often. So think of a bacterium, which is very, very small, right? It's in the scale of nanometers, which is, if you take a meter and you divide, 1/10 of it, then 1/10 of that, 1/10 of that, multiple times over. So we're talking about very, very small creatures. So they also have very little bits of DNA, which means you have to find a way to make many more copies of that DNA, right, each one, and then read them in a machine. And so the costs of doing this have reduced. We've become better and faster at doing the sequencing. And as a result, even though we can't see the bacteria, we can't culture them always, we can still sequence them. And that is a huge revolution that has happened in biology in the last few decades. And the reason it's exciting is because now we have our hands on all of these data about which species there are, how many species, what are they doing, how different are they from each other, right? So So with all of this data, we've also had to have new ways of processing this information, right? Earlier we were dealing with, okay, you're sick, you have maybe a staphylococcus infection, maybe your child is sick, maybe he's crying a lot because his ear is hurting, maybe he has an ear infection. Let's find the one bacterium, right, typically, that's causing this problem and fix it. But now we're finding actually, now that we have access to these data, are the groups of bacteria are what are turning out to be very important, right? And we're still trying to figure out why and how are they important, right? Instead of just focusing on single species. So we need new methods then to also process this information, to condense it, because our brains can't process this kind of data, right? So that's also leading to another revolution within this field. And I just want to say that a lot of this has involved cataloging, right, in some sense. So just saying who's there, what are they doing, how many of them are there, right? Which bacteria are more abundant than others, and where are they more abundant? What makes them more abundant? So a lot of this initial process is simply saying cataloging, right? Like what the British did across the world wherever they colonized. They would go and make very nice notes about, which beetle is where and how many of them do they find. And as a result, we know a lot about the animals and plants in so many parts of the world, right? And we have continued to do this, and we continue to do this a lot. But we've done this particularly in my lab with bacteria associated with these groups of insects. Dragonflies, you guys have seen dragonflies? Yeah? Butterflies and beetles as well. And so I won't talk too much about this, but I just want to say that this cataloging is also important because without it, you can't move ahead. But now, once you have a catalog, you can start asking really fun questions about why that matters, and does it matter, right? Because it may be that because the dragonfly is eating other dragonflies or eating other butterflies, it happens to get some bacteria in its belly, and they're there, some of them survive, but nobody cares about it, right? The host doesn't care, the bacteria are just there, fine. Tomorrow there'll be a different bacterium if it eats a different food, it's okay. So is that the case? Or are there very specific associations between the bacteria and the insects or animals, other animals in which they're living, right? That's the kind of question that we can start answering now. Okay, so I think my title, if you notice, said something about enrichment, right? So how are we enriched by bacteria? All of this stuff is fine, there are bacteria and so on. But why should we care? Why is this enriching? So I'm going to give you just a glimpse of three ways, more or less, in which I think bacteria are enriching us. And by us, I mean not only us humans, but also many other animals, plants as well. I don't have many slides about plants, but I'm happy to discuss if you like. Okay, so here's one kind of way in which they can enrich, right? That it's about the sheer numbers. If you think of richness, what comes to mind, perhaps in some senses, what is the big pot of money I have in my house or in my bank or something, right? So numbers matter. So here are some numbers just to give you a scale. So an average human being has more than 10,000 types of bacteria in his or her body, okay? Most of these are actually in our gut. So imagine 10,000 types of little wriggly things in your belly. That's a lot, right? So the question is, what are they doing? How many of them are there? If you take the weight of all of these cells in your belly, they add up to 1.5 kilos, right? And this is just to give you an idea of what that means. This is a bag of flour, right? That's a lot of bacteria, right? You think about it. Also, the total number of cells in our body, about 60 trillion. 30 trillion are bacteria, and 30 trillion or so are our own cells, human cells. So, and then of course, none of you will be surprised if you find out that similar proportions are reflected in your poop. So when you poop, half of your poop is about bacterial cells, right? And this is not even counting viruses and fungi that are also living with us, on us, inside us. So once we got to know about some of these numbers, it's sort of staggering, right, to think. So what does it mean when I'm saying I'm I, or you are you, right? What do we mean when we say that, when we are housing so many of these things, right? And so these are questions, again, that are very interesting, to some extent philosophical, but also important, because then our identity, our self, is how we relate to the world and is how we evolve as well, right, as entities. So there are now people who are thinking about how, in terms of evolutionary biology, how can we better understand these interactions? Are there interactions between all of these 10,000 types of bacteria in our body? And how do they matter to us? Any questions, observations, comments? Oh. Yeah. Oh, good point. So we chose butterflies partly because butterflies have very interesting life cycles, right? So they go through larvae that eat largely leaves, so very solid food, and then the adults are feeding on nectar or plant sap and dung, animal dung fluids and so on. So it was a very big change in their diet, right? And so if there is a role that bacteria play in their nutrient absorption, which we know happens for many insects, it would seem very dramatic that there should be a big shift in the bacteria as the butterfly grows up from a caterpillar. So we wanted to ask if that happens, and if so, what are these bacteria doing? Because when we started this project, nobody had really studied bacteria living with butterflies. And we thought that's a great system to work with. So that's why we chose butterflies. We chose dragonflies, honestly, partly because dragonflies are just so cool that I wanted some reason to work with them. But also, they are predators, right? They're top predators in aquatic ecosystems where they're zipping around. You've seen them, right, catching insects and so on. And it seemed like they, therefore, would then assimilate all of the microbes from the environment because they're eating so many kinds of insects, right? And they're thought to be sort of what's called generalist predator. So they're not particularly peculiar about what they'll eat. Anything they can catch, they will eat, right? That's the logic. But what was really fun was that we found if we sampled the same species of dragonfly from our campus versus in Agumbe versus in Shendurni versus in Nagpur, Assam, they all had very different kinds of gut microbes, right? Bacteria in their guts. And that was surprising because you think the same species should eat pretty much the same stuff everywhere, right? It doesn't. And then what we found is, again, if we use sequencing to figure out what their diet was, because observing dragonflies is so hard, it's very hard to say what they captured and what they ate. So we have very little data on what dragonflies actually eat. I mean, each dragonfly eats. So, but we can use DNA to figure that out because we can sequence what's in their gut, right? And we can say, okay, it's a fly, it's a butterfly, it's another dragonfly that they ate. So we did that, and we found that actually, depending on the location, depending on the season, depending on the species, dragonflies are actually eating different things. And that explains, potentially, why the microbes in their gut are different. Yeah. So basically, it was an attempt to start learning more about wild insect populations because we know a lot about fly populations in the lab, which is what people typically study. And so we started doing this. And especially in India, there was absolutely no idea what was there. So we thought we'll make progress. Yeah.
In the case of humans, is this prenatal or is it after birth? Like that?
Yeah, so the gut bacteria actually, when a baby is born, depending on how it's born also, it makes a difference. So if it's C-section versus normal delivery, typically babies will have different numbers of bacteria because they acquire them through the womb and through the passage as they come out. But as babies grow, maybe up to 1 to 2 to 3 years of age, their gut flora settles down very quickly. And then you have what's a typical adult flora. So babies will have much fewer bacteria. And actually, there's a very interesting story, I'll come to it later, about it. So, but after they're weaned from breast milk, their gut flora changes. And then after 3 years of age, unless something majorly dramatic happens that changes the baby's diet or growth in some way, the flora stays fairly constant throughout its life. So it settles down very quickly. But it's lower for a baby, yes. Good question, yeah. So the question is what happens with insects? Is it similar to human babies? It differs in different insects. So in some insects, what's really cool is when they depend on a specific set of bacteria for their growth or development, which they do, right? They will pass on, the mother will lay, in some cases, extra eggs, okay, which are not really fertilized eggs, but they're just sort of fake unfertilized eggs. But the eggs contain a shot of bacteria, right? And so the caterpillar comes out and eats that extra fake egg, gets all the bacteria, and it's set. Right? So there's sometimes that happens. In some cases that doesn't happen. So in butterflies what we found is it didn't seem like the mother was passing anything to the offspring. But the larvae, as they started chewing the leaves, would basically just get bacteria from the leaves, and that's what we found in their guts, more or less. So that's something also interesting that we found, is that they didn't seem to depend on bacteria, which I'm still very surprised by, honestly. We haven't quite figured out why or how, because the expectation in general was once people started finding all of these microbes that are super important, right? The expectation is that every time you're eating something, right, you need to sort of depend on microbes maybe. So people have been searching for this signature a lot, and we failed to find it for butterflies at least. So it's still a bit of a curious mystery how butterflies, all the different species of butterflies that you've seen, there are caterpillars, right? And you must have seen them, that they're eating so many different kinds of plants, and they're a major pest in some cases. But they don't seem to care about the microbes. They don't seem to depend on the microbes. Whereas you and I, we all depend heavily on the microbes in our gut. And I'll talk about that a little bit in the next few slides. Any other comments, questions? So we found dragonflies, depending on the species and where they are and the season, they eat flies, they eat other dragonflies, they eat butterflies, they eat sometimes some bugs. So they eat many different things. So that's one reason I gave you. Another reason is that if you have the right set of bacteria, they can be the good guys, right? And they can sort of prevent the bad guys from establishing. This is what happens in movies you've all seen, right? There's a villain, there's a bunch of villains, and our hero has a hard time because there's too many of them. So in essence, this is what's going on here, right? So I'm going to give you the case of this particular bacterium called Bifidobacterium infantis. And it's a long name, but focus on the infantis part, right? The reason it's named this is because it's as the paper said, champion gut colonizer. Right? What it does is it's found in babies' guts, in infants' guts, and increasingly it is being lost from Western populations, right? So Western countries, babies don't seem to have this bacterium very much, but most other parts of the world, they seem to still have this bacterium quite a lot in the infants. What's special about this bacterium? What it does is if it colonizes, it prevents other bacteria from getting in, right? So pathogens, whatever other bacteria, which is not so great. And the reason this bacterium is fantastic, so this links to another puzzle that people had been thinking about for a while, which was that if you look at the composition of breast milk, right? Breast milk has a lot of very complex sugars, okay, which babies don't have the enzymes to digest. So what are all these complex sugars doing there? Because they're very hard to make. You know, the mother has to spend a lot of energy to make these fancy sugars, put it in the breast milk, and then what's the point? And then they found that the point is to feed this bacterium. Okay, so this bacterium is one of the few that has the ability to break down these sugars, and then it allows it to colonize the gut. Once it's colonizing the gut, it of course provides nutrition for the baby because it now breaks down these complex sugars, gives them to the baby, and it prevents other pathogens from colonizing, right? So the story is really, really, I think, nicely links up, right? And it's a great example of how even when the gut microbiome is very narrow in some sense, not too much diversity, not too much complexity here, it's still very, very interesting things are happening. And then of course as the baby is weaned from breast milk, starts eating normal food, Bifidobacterium infantis will decrease and go away after some point. Yeah. So I want to talk about some experiments that we have done in our lab recently, which allow us to observe some of these relationships changing in time, right? And that's why I find this very exciting. So let me tell you a bit about this beetle. This is Tribolium. It's a flower beetle. If any of you have seen flower in your house, which you've forgotten for some time, it'll have some small insects in it, right? It's a very small beetle. There'll be many kinds in your flower. So maybe you have seen Tribolium, maybe you've seen a different species. Doesn't matter. The point is they're all pests and they're very annoying. But they make for great model systems in the lab, right? Because they reproduce quickly, they're easy to handle. You know, all you need is some atta and that's it. So, so we have these flower beetles as a nice study system in the lab, and they're usually eating wheat flour, okay? And the wheat flour itself has some bacteria associated with the flour. because when you harvest the grain, you get some bacteria that are associated with it, right? So I'm just going to show you these bacteria here as pink one and a gray one. Okay, the community actually is more complicated, but I think this serves the purpose. So now what we do is if we give these to beetles, and in one case we wipe out all of the bacteria, okay, and we give females some time to lay some eggs. What we find is that females will lay many more eggs when the bacteria are present, but if we remove the bacteria, they don't lay as many eggs, right? And so somehow they're sensing that are there bacteria in the flower, and if so, I will lay more eggs. And we have no idea yet why or how this happens. Okay? But what is clear is that they depend on the bacteria in the flower. Okay? So now this poses a problem, because if you put them in jawar, which is something they normally don't eat, okay, or don't eat very well, they don't infest jawar so much. So you almost never will find these beetles in jawar in your house. Doesn't matter how old it is. Okay? Jawar has a different set of microbes, right? So here I'm just showing you yellow and gray. There's some overlap, but there's some differences. And now if we do the same experiment with females here, what's really interesting is that if you wipe out the microbes, they lay, in both cases, they lay fewer eggs. Okay? So in jawar, they don't seem to care about do you have bacteria or not. But now that presents a problem, because then how is this beetle going to establish or colonize jawar? I don't want it to, for all of our sakes, but in general, as a principle, right, if you're establishing in a new environment and you are dependent on your microbes in your old environment, and that's how you get your microbes somehow, what do you do when you're moving to a new place, right? So all of you have traveled in trains at some point. Many of you will have your dabba, which you carry with you, right? Make sure that doesn't matter where you're going, you have some good food to eat. So what does this bug do? So we asked this question in the lab, and we allowed the beetles to evolve, okay, in the lab conditions, where we keep them on jawar, they lay some eggs, the eggs grow up into beetles, again the beetles are allowed to lay eggs and so on. Right? And what we're doing is, by only choosing beetles that manage to become beetles in some shorter time frame, we basically impose strong selection, right? We force the population to make beetles that are better able to survive in jawar, right? And so now what happens with time is, let's say in cycle 1 there were few beetles, few eggs. Cycle 5 we have more beetles, more eggs. By cycle 20 everybody's looking quite happy. Yeah. Nice beetles, nice eggs. Now what's really interesting is if we look at the microbes, you remember they started off with yellow and gray, they end up with pink and gray, which is what they had in wheat always. Right? This is really fun, because now what this suggests is that if you are going to new habitats and you carry in your gut your nice microbes, over time those may establish in the habitat and you're back, you're fine. And what has happened is that in association with these microbes, potentially, we don't absolutely know this yet, we continue studying this, these guys have become better at colonizing jawar now. Right? I promise not to release these in your houses. But this is a really nice example of how very quickly, 20 cycles is about 2 years of our 2 years. That's nothing. Right? And maybe this kind of phenomenon explains why many insects are such great pests. Right? They just quickly transition to whatever you come up with. And there are many good examples of this. I can talk about this later. Yeah.
Basically, we were talking about the microbes that were in our gut right here. So in that, let's say we have a certain intestinal disorder, and we need those useful microbes, right, to process our food. But during that disorder, a lot of them are washed away, and we need supplement. Is there any way that you can code our DNA so that we ourselves can produce those microbes and we don't need any supplements to that?
Produce the microbes?
Themselves, like the.
Or you mean the kinds of genes?
The bacteria.
Yeah.
The lactobacillus.
Yeah. So it's hard to, just to be clear, it's hard for us to make bacteria, because that's basically like one species making another species. That would be very hard. But in terms of the genes being transferred in our genome and us being able to produce some of these critical vitamins or something, in theory, yes, it's possible, right? In practice, it will actually be fairly challenging, because a lot of these vitamins and amino acids that we rely on getting from our diet or from bacteria are complex. You know, there are many, many pathways that are very sort of like this. So you'd have to move all of those genes over. Another sort of practical problem, logistical problem also is that the way that bacteria encode genes and express genes is a bit different from what we do. That means that all of those genes may not do as well or work as well in our cells. So there are some logistical challenges. And then, of course, there are ethical challenges involved about editing our genomes to mess around with what we are not able to do. But in theory, yes, possible. So I think that's it. That's all I have in terms of slides. I'd like to leave you with the impression that we live in a world of microbes. And if we have to have any handprints or footprints in the future, we have to think about microbes a lot more. I'd also like to thank my group. This is my team of people at NCBS, all of the various sources of funding that have supported the work I showed you.